When the genetic code in healthy cells is damaged, these cells are safely removed by a process of programmed cell death called apoptosis. You can think of this as a fail-safe mechanism, which, as you can imagine, is extremely tightly controlled. In cancers, when cells become damaged, they don't die. Instead, they continue to grow and divide, passing on the damaged genetic code. These cells are resistant to apoptosis. In the treatment of cancer, resistance to apoptosis is a significant barrier to the efficacy of many chemotherapeutic drugs and radiation therapy, which aim to induce apoptosis in cancer cells. Therefore, targeting apoptosis has become an attractive strategy, but has been hindered by a critical lack of understanding of the regulatory processes and proteins involved. In the search for a cancer drug, I'm targeting the interaction between BMX and BAC. When BAC is activated, it irreversibly permeabilizes the outer mitochondrial membrane, causing the release of apoptotic factors which commit the cell to death. Drugs which activate BAC have been developed, but have been found to have on-target toxicity, which has resulted in limitations in their use in the clinical setting. Therefore, there's still an unmet need for drugs which induce apoptosis without causing significant side effects. I discovered it's not enough to use drugs that activate BAC. BAC needs to be in the right state to become activated, and BMX controls this state, preventing BAC activation. My approach is to inhibit BMX to resensitize cells to existing chemotherapeutic agents, so that lower, less toxic doses can be used. We have substantial data to support the BAC-BMX interaction. I've been studying the regulation of BAC using cell and molecular biology techniques for the last 10 years and have a deep understanding of the regulatory mechanism. Working in LISB, I've built expertise in crystallography and cryo-electron microscopy and have access to state-of-the-art facilities and expertise which will boost my drug discovery efforts. This combination of biological knowledge and the ability to direct an atomic-level search for the right drug candidate is not held anywhere else. Soon, we will be testing the in vitro functionality of molecules which were identified in silico screening in our panel of physiologically relevant cell-based assays. We will also be testing these molecules for efficacy in combination with the relevant standard of care treatment regime for breast, prostate and non-small cell lung cancer cell lines. We are looking for industrial partners with an interest in repurposing existing agents for use as dual therapy and overcoming acquired resistance to convert this proof of concept into a candidate therapy or platform technology.